Welcome, welcome. This is Classic Dominance Mendelian Genetics Dihybrid Crosses, 4 pages 5 to 6 on that document if you want to follow along with me. I have it printed out on paper. I think it's easier for me to explain what's going on as I'm writing it versus watching me just kind of like type stuff. Not as helpful. So let's get started. Problem number 1. In rabbits, gray hair is dominant. All right, so I'm going to get out my colors here. Gray hair is dominant to, let's use some purple, is dominant to white hair. Also in rabbits, we're going to use even more colors. Black eyes are dominant to, let's use red eyes. All right. The following letters represent the genotypes of the rabbits. So gray, capital G, is the dominant fur color. White, little g, is recessive in fur color. Okay, great. So gray is big G. White is little g. And looking at our eye colors here, black or big B is dominant to red or little b. So they've given us letters. We don't have to think about it. Tells us what is dominant, what is recessive. So great, we can move on to the next piece. Part A, what are the phenotypes of the rabbits found with the following genotypes? So here's some practice using, applying our knowledge of phenotypes and how they relate to the genotypes. So the genotypes will tell us what the phenotype is. Let's look at some of these examples. So first up, we have big G, little b, little b. All right, I'm going to break it up here with our colors. I did orange and orange for, got it. All right, so here are all of my colors. Awesome, awesome. So we have a big G, which is orange, and we have a little g, which is purple, and little b, which we decided would be red or pink, I guess I could say. Little b. All right. Well, knowing what we know about dominance and recessiveness, if there is a big capital letter or a dominant gene for that specific trait that we're looking at, in this case, the color of the hair, that will be dominant. So let's separate these for a minute. Hair color versus eye color. All right. So here we got a big G, meaning it's going to be dominant. So we'll be gray. Ooh, it's awkward to write with pencil on my wrist. All right, and then let's look at the eye color trait here. We have little b. That means it's recessive. Homozygous recessive will give us the recessive phenotype of red eyes. All right, let's move down one here. Great. So we're going to separate these a little bit so we can look at our two different traits. We have G's and B's, two little g's. That means purple and purple. And two little b's, which means little b. This one's pretty easy. It's going to be recessive for both. How do we know that? They're lowercase. So little g gives us white hair. And little b gives us, we already found out in the previous one, red eyes. All right. Now let's move over and work on these practice ones here. Little g. So those will be purple. I'm not going to separate these so we can get used to it. And big B. Big B is black. So they will have black eyes there. It's just the green color. I know it's getting started to get confusing. So what does our phenotype look like? Well, these little g's, because there's no dominant allele for our hair color, the recessive color will be present. We found that out in this one right here. So it will have white hair and black eyes. How do we know this? There's at least one dominant allele when we have two big B's. So it will have black eyes. All right, last practice one here. Big G, which is orange. Little g, which is purple. Big B, which is green. Now I'm flipping through my pens. And little b, which is pink. Ooh, looks like we have double heterozygousness here. Well, what does that mean for our phenotype? What traits are being expressed? What color is the hair? What color are the eyes? Well, looking at just our G's here, there's at least one dominant allele. We know that the dominant color is gray. So this bunny will have, oops, gray hair. And looking at our eye color here, separating it out, the B's, we have a heterozygous, meaning we have at least one dominant allele, that big B, giving us black eyes. So when we're looking at these dihybrid crosses, we need to sort of separate out the two different traits that we're measuring. One is for hair color, one is for eye color in this particular example. All right, part B. A homozygous black-haired, red-eyed male is crossed with a white-haired, black-eyed female. What does that mean? Oh, I didn't write. Okay, I'll make this change on yours. We're going to say that it is a heterozygous black-eyed female. All right. So B is asking, what are the genotypes of the parents? So we're given the phenotypes, the hair color, the eye color, hair color, eye color, but what are the genotypes? What does that look like with the letters for our alleles? All right, so let's take a look at the male because the male will come first. Homozygous black-haired. That's not a thing. I had to change that too. I'm so sorry. I did not read this all the way through. Homozygous gray hair. Glad I'm finding it now so you don't get confused when you do this. If you like the practice. All right, again, I apologize. Homozygous gray-haired, red-eyed male. Okay, so gray hair is dominant. Oops, I can't see. Gray hair is dominant. And that is signified with a capital G. And if he is homozygous, that means they're going to be the same capitalizations. So he will have big G, big G. And red-eyed. Well, what does red eyes look like? Red eyes are recessive. Let me get my pink one out here. And in order to be recessive, we need them to be homozygous. So little b, little b. Great. Now let's look at the female. She is white-haired and heterozygous black-eyed. Cool, cool, cool. So white hair, what does that look like? White hair is recessive. And in order for it to be recessive, that means her genotype must be homozygous recessive, little g, little g. And she's a heterozygous black-eyed. So black eye is dominant, big B. Heterozygous means one of each, big B, little b. She'll be big B, little b. Just try to keep with my color coding here. I apologize if that is confusing to you. All right, great. So we figured out our genotypes here. I'll even write them in all black just to make perfectly clear. Big G, little b, or G, G, B, B. All right. Now figure out what we need to put in the pair boxes in our Punnett square using the FOIL method. So looking at our Punnett square here, we need 4 options for each parent, and we're going to find that out by foiling. Foiling. So first, outside, inside, last. For the male, we know what his genotype is. We got to FOIL these guys out. Okay. So first, let's use the color I have been using. Awesome. So first, we got to use the first letter for each of these 2 genes. Big G, little b. Great. Now we did the first, now we need to do outside. So what are the 2 outside letters? Big G, little b. Great. Now let's do, what's next? I, for inside. What are the 2 inside alleles here? Big G, little b. And the L in FOIL is for last. What are the last two of each of these traits? Big G, little b. Awesome, awesome. We did it. We FOILed for the male here. Next up, the female. We're going to have to FOIL all over again. So first, what are the first two letters of each of these traits? We got a little g and a big B. Now we're going to pause. Our best practices usually tell us that the capital letter needs to come first. However, when we're talking about dihybrid crosses, we need to be consistent about our letters before we can worry about our capitalization. So since the G's are always coming first, this little g is going to come in front of the capital B. I know it feels weird, looks kind of crazy, but that's just the best way to keep track of all of your alleles. As weird and gross it feels to put the lowercase in front of the capital letter. All right, so we did our F. Now we're going to move to the O, the outside. What are the two outside letters here? Little g, little b. Awesome, awesome. All right, now we need the I, the inside, the two inside terms, two inside letters, little g, big B. And the L for last. What are our two last letters of each of our two different traits? One G, one B, both little. We did it, we foiled. Awesome, that was like the hardest part. In my opinion, the foiling is the hardest part because even I get it messed up sometimes. Okay, now moving down, we're going to make our Punnett Square. So we figured out what needs to go in our boxes across the top and down the side. Then we're gonna fill it in. So the male, generally, best practices say male goes across the top, female goes down the side. So since the male is first, that works out just great. Happens to be that all of his are big G, little b. Easy peasy. I'm going to use, gosh, black for the male. All right, I committed. Big G, little b, all the way across. Perfect. And for the female, let's use a new color. Let's use a dark green. How does that sound? Dark green. Make sure it works. Awesome. All right. So we know what that's going down the side for our female here. So first is little g, big B. Next is little g, little b, little g, big B, and little g, little b. Oh, you can barely tell the difference. That's okay. Now we're going to combine. We're going to cross our two parents. Now remember what I said up here, letters are priority. So the first thing we need to do is make sure all of our letters are together. The G's are with G's, the B's are with B's, like we saw up here. Then within each letter, we can look at the capitalizations. So first up, let's do even another new color. I have so many colors, thanks to my husband. All right, let's try this light purple. It's not even used yet. See what that looks like. Ooh, that's kind of pretty. All right, sorry, I love colors. Here we go. We're going to look at the G's first because they're coming first. We get a big G from dad, so the big G will always come before the little g. So we got a big G from dad, and looking at the G from the mom, we got a little g there. And the B's, I'm gonna use, oh man, I'm using so many new colors, I'm gonna run out. Gosh, I may have to reuse a color. All right, I will reuse orange. All right, so for the B's, we have one big B coming and a little b coming. Big B needs to come first. So big B from mom, little b from dad. So please notice, letters stay together, and then within the letters, we worry about the capitalization. Uppercase comes before lowercase. All right, moving on. Next one over, sometimes I like to go across, sometimes I like to go down. Today, I guess I'll just go across. There's no right order to do it. If you like to go diagonal, it's kind of cool too. All right, so for the G's, we got a big G from dad, and a little g from mom, keeping my g's purple. Big B from mom, little b from dad. And it looks like since dad is all the same across the top, all of our rows are going to be the same across as well. All right, so we got a big G from dad, little g from mom, big B from mom, little b from dad. Big G from dad, little g from mom, big B from mom, little b from dad. Hey, we did it. We did our first row. Awesome. Now, it's not going to be exactly the same because mom has a different B allele in this row. But let's focus on our G's first. G's come first. All right, so we got a big G coming down from dad, little g from mom. Now, little b from mom, little b from dad. Ooh, that's different. That doesn't look like the row above. Now, since dad is the same, our entire row is going to look the same here. Big G from dad, little g from mom, little b from mom, little b from dad. Big G from dad, little g from mom. These pens are weird. They're not rolling smoothly. Little b from mom and dad. Big G from dad, little g from mom, little b from each parent. All right, moving along here. This one is going to look just like our first row because our genotypes are the same here and our genotypes are the same there. So big G from dad, little g from mom, big B from mom, little b from dad. And repeat all the way across. Loving Punnett squares. In my opinion, once you get the hang of it, it's actually quite fun. All right, now last row. Looking up here, it's going to be exactly the same as this one up here because dad stays the same. Not all Punnett squares are going to be this easy, I promise. But this one just happens to work out in kind of a nice way. All right, so big G from dad, a little g from mom, little b from each. Big G from dad, little g from mom, little b from each. Big G from dad, dad, little g from mom, little b from each. And the last one here, big G from dad, little g from mom, little b's from both parents. All right, cool. Tedious, yes, but not the worst. We did it. We figured it out. All right, now phenotypic ratios down here at the bottom. Let's scooch up a little bit. All right, how many are black for? So I do that again, and let's change that. It's gray and white, black and red. All right, gray. Wait, okay. Sorry, I will make that change on yours. All right, getting into it. I'll go back and use my pink color. So how many are gray with black eyes? Well, gray is dominant, so wherever we see a big G, looks like all of them, we're going to have a gray rabbit. So right off the bat, we know since there's a big G in every single one of these offspring squares, we're going to have none that are white. And since these last two counts here are going to be white fur, we know none of them will have that white fur because we see this big G in all of our squares. Now, how many of them are gray with black eyes and how many are gray with red eyes? Let's take a look. Let's use some colors here. How about yellow and blue? Yellow and blue. All right. So gray and black. Gray for black eyes. Black eyes are dominant. So wherever we see a big B, that rabbit will have a black eye color. So wherever we're going to see this, at least one big G and at least one big B, that one will be, I'll make this yellow. Going across, there's at least one big G and one big B. Here we have all little b's, so none of them will be black-eyed. And this one also has at least one big G and one big B. Great. Now, gray fur with red eyes. All of these have gray fur, easy enough. Red eyes is recessive. So if we see a double little b, homozygous recessive, we know we're gonna get red eyes. So let's take a look here. This one is gray with little b's. It's going to be gray with red eyes. Now we just count them up. We have 1, 2, 3, 4, 5, 6, 7, 8 that are gray with black eyes. And 1, 2, 3, 4, 5, 6, 7, 8 with red, gray fur and red eyes. Just to double check, we should have 16 of these squares. So we should have a total of 16 in our ratio here. A plus 8 gives us 16. We're good to go. We counted all of our offspring correctly. Okie doke. Now let's move on to problem number 2. Problem number 2. All right, let's push this down so we can move the blur. All right. Excited I get to use lots of colors again. Imagine that a couple is planning to have children. The ability to roll one's tongue like a taco is dominant, big R, over the non-rolling condition, people who can't, little r. Unattached earlobes, which I happen to have as well, interesting. Unattached earlobes, capital U, are dominant over attached earlobes, little u. Okay, lots of information. Let's break it down. So, rolling the tongue is dominant with a big R. Let's use pink. Non-rolling is a little r because it's recessive. If it's not dominant, it's going to be recessive, and we're talking about classic Mendelian genetics. All right, then we're going to have unattached earlobes are dominant, and attached earlobes are recessive. Great, we figured out what's dominant, what's recessive, what letters to use. Now moving on to A. The male is heterozygous for tongue rolling and homozygous dominant for unattached earlobes. All right, let's get out my colors. Rolling the tongue is dominant. He can do that, but he is heterozygous. That means he's going to have one of each capitalization. So one big R for rolling the tongue, and when I use pink, one little r because he is heterozygous, and he is homozygous dominant for unattached earlobes. Great, that is big U, big U. Homozygous dominant, homo means the same capitalization, dominant means capital uppercase U. All right, and the female is homozygous recessive for tongue rolling. So we know that non-rolling is recessive, so she'll have little r, and she is heterozygous for unattached earlobes. So heterozygous means different, one of each capitalization, she'll get a big U, and because she is recessive, she'll get a little u as well. So she is heterozygous, one of each capitalization. Just to make things a little less crazy here, I'll also write these out in black. R, R, U, U, R, R, U, U. All right, so now we know what our parent genotypes are going to be. We need to figure out what we're going to put in our parent boxes. Looking at our parent boxes here, we need to FOIL. We have 4 boxes to fill for each of our parents. Starting with the male, we know he is RR heterozygous, UU homozygous dominant. We got a FOIL. So first, what are our first 2 terms of each of our, move this down a little bit, of each of our traits? The first one, for tongue rolling is big R. The first one for earlobes is big U. Great. Now we're going to go to, outside. The outside two terms, big R, big U. Inside, our two inside terms, we have a little r and a big U. And L for last, what is our last two terms of each of our traits? A little r and a big U. Cool. Now let's look at our female. Our female, we need to FOIL her as well. So first, the first two terms of each of our two different traits. So she has a little r for tongue rolling and a big U for unattached earlobes. O for outside are two outside letters here, a little r and a little U. Inside, what are our two inside letters here? Little r, big U. And last, what are the last two letters of each of our different traits? One here is a little r, the other one is a little U. R, U. I tried to exaggerate the big and little because my U's look exactly the same except for their size. Now we FOILed. That's the hardest part in my opinion. Place those FOILed letters here in the top and along the side. Let's go back to orange. We'll use that once. All right, so the male, just transfer these over here. So we have big R, big U, big R, big U, little r, big U, little r, big U. Great. And let's also use light green because we'll use that once before too. So for the female, plug these in down along the side. So little r, big U, little r, little U, little r, big U, little r, little U. Awesome, awesome. Now, what other colors are we going to use here? We haven't used blue yet, so we'll use blue and black, I guess. It's kind of boring. All right, black and blue. Here we go. So we want to do our R's first. So we got big R from dad, little r from mom. Which one goes first? The capital R goes first. So big R from dad, little r from mom. And then our U's, both of them are capital. One from mom, one from dad. Let's just say, since we went across last time, let's go down this time. All right, so we're going to get another big R from dad, a little r from mom, a big U from dad, and a little U from mom. Continuing down, another big R from dad, little r from mom, and two big U's, one from each parent here. And lastly, our little RU here and a big RU there. Big R from dad, little r from mom, big U from dad, little U from mom. Hey, we did our first column. What's kind of cool is that now we know that this one is the exact same genotype as this one. So we can just copy down what we just wrote. Easy peasy. Big R from dad, little r from mom, big U from each parent. Big R from dad, little r from mom, big U from dad, little U from mom. Big R from dad, little r from mom, big U from each. Big R from dad, little r from mom, big U from dad, little U from mom. All right, now our dad genotype is changing. We got to pause. We got to do it slowly, and then we can repeat it for our last column. All right, so we don't have either of these giving us a big R. So we're going to have little r, little r. Looking at our U's, great. We're getting two big U's, one from each parent. Next up, little r from dad, little r from mom, big U from dad, little U from mom. Two little r's again, little r from dad, little r from mom, big U from each, big U, big U. And last one in our column here, little r from dad, little r from mom, big U from dad, little u from mom. And once again, since we know dad is going to be the same, we can just copy it over the way we had it before. So little r from dad, little r from mom, big U from dad, big U from mom. Little r from dad, little r from mom, big U from dad, little u from mom. Little r from each parent, big U from each parent, and lastly, little r from each parent. Oops. Big U from dad, little u from mom. Whew. All right, now our phenotypes. Get out my colors. I've got lots of colors here, ready to go. So let's just go ahead and assign some colors here. I'll make rolling tongues and unattached earlobes green, rolling tongues and attached earlobes pink, non-rolling, no, I'm not going to use blue, I have the blue pen, it might disappear. Non-rolling and unattached yellow, and non-rolling and attached in orange. Okay, so rolling and unattached. What do we know about that? So rolling is dominant, unattached is dominant. So if we ever see a big R and a big U together, it's going to be this green color. So here we have a big R, at least one, and it's going to be dominant expressed. And we have at least one big U here. Great. Perfect. It's going to be this green color. Next one, big R, at least one big U. Green. At least one big R, at least one big U. At least one big R, at least one big U. That whole column there is going to be this green rolling unattached. Moving over. At least one big R, at least one big U, another set of green. At least one big R, at least one big U, another green one. At least one big R, at least one big U. Same for this one as well. Okie doke. Now, moving on to this column, I don't see any big R's. No big R's. So what does that mean? It means we're going to be non-rolling. And we need to keep an eye on those U's. So unattached is dominant. So let's see what we got here. So we got little U's. We know they're going to be non-rolling for these two. So it could be yellow or orange. I'm going to get rid of my pink because I know it's not going to be pink. None of them are rolling and attached. We don't have any pink here. So right off the bat, that's going to be a 0. Now we have to determine which ones are going to be yellow and which ones are going to be orange. Which one's going to be orange, which one's going to be yellow. So we know that it's going to be non-rolling because we have these recessive R's here. But now we have to think about the attachment of the earlobes. Are they unattached and dominant, or are they attached and recessive? Well, looking at this first one here, we have at least one big U, meaning it's going to be yellow. Well, this one we have at least one big U. This one we have at least one big U. This one we have at least one big U. All of these have at least one big U. Bye, orange. So we know that there's no orange on here either, no orange highlighter. Now we need to count them up. 1, 2, 3, 4, 5, 6, 7, 8 that are green. 1, 2, 3, 4, 5, 6, 7, 8 that, oops. Yes, 8 that are yellow. Do we add up all of our offspring? There should be 16 total offspring. 8 plus 8, that gives us 16. We counted all of our offspring. All right, that brings us to the end of our dihybrid crosses. I hope this was helpful.